Hello. We are back. The Lecture 3 series, or kind of week 3 of this type of material, Extracellular Matrix. This is Chapter 5 in the textbook, if you're reading the textbook. We're talking about the Extracellular Matrix, aka the ECM. Ooh. Objectives: function composition of the extracellular matrix, the different collagen fibers, ground substance, adhesive proteins, integrins, structure and function, and then composition function of basal lamina. A good amount of these kind of things that we're going to go through in this PowerPoint, we've actually already touched on previously. So some of the stuff like the integrins, adhesive proteins we covered previously. Basal lamina we touched on a little bit. Collagen, super briefly touched on it. But so a lot of these terms you should see these pictures as well, should seem familiar because they're coming back. So extracellular matrix, looking all in through here, right? We have those gel-forming polysaccharides, collagen, fibronectin, integrin, all these kind of stuff. We talked before about the plasma membrane. We talked about cytoskeleton, all of this kind of stuff. So, with the extracellular matrix, major functions. One of the biggest one is a reservoir. A reservoir is just a fancy way of, I feel like I'm saying that weird, reservoir. Reservoir? I don't know. Something like that. No, you're never going to forget that word. Reservoir for water, ions, nutrients, hormones, cell signal molecules, etc. Stores stuff. Okay, a reservoir is what, you have a plentiful of it, you're going to store it, you're going to hold on to it until you need it. The other functions: maintain strength and rigidity of tissue so we don't turn into a big old pile of goop. Maintain the shape of the tissue. All that's kind of similar. And then the medium through which the nutrients and metabolic wastes are exchanged. Medium just referring to like the stuff, the material through which you're going to exchange nutrients and the waste products, which we'll get into a lot more of how that actually happens in Phys 1 next trimester. When we look at the components, we have structural proteins, such as collagens and elastins. We have the protein polysaccharide complexes, those proteoglycans, which we've seen before. And then the adhesive glycoproteins, known as fibronectins, laminins. There should be another comma right there. Fibronectins, laminins, and integrins that bind and attach stuff together. Know all of those words. That's what we need to know. Again, down below, I've taken the time, sorted it all through a bunch of these, given straight definitions, kind of what each of these is, kind of what's going on with that. So look in here. This is just the example. This is the image of our extracellular matrix. And this is called a loose extracellular matrix, composed mainly of molecules of the ground substance. That's because this is an embryo. So with an embryo, we're going to have a loose extracellular matrix. Primarily fibroblasts. All these here, fibroblasts are the cells that are actually going to be creating the connective tissue. Anytime you see that word blast at the end, blast, you'll see it at the end. So blast equals builder. So like you'll see osteoblast, that is a bone builder cell. On the other side, you'll see clast, that is a breakdown type cell. So like that, you have osteoclast, that equals a bone breaker downer. That's not real words, but you get the idea. Blast is a builder, so fibroblasts build cells. We have this stuff called ground substance. Okay, so ground substance of the extracellular matrix. Let me actually get a specific. The ground substance, make sure I get a good definition for you. I'm going to throw this into here. So the ground substance is an amorphous, so it doesn't have a full shape, kind of just oozes into everything. Gel-like substance in the extracellular space of animals that contains all of the components of the extracellular matrix. Okay? So the ground substance is kind of that goo that's in between everything. It's highly hydrated, found with a bunch of water. We've covered that a couple of times. Our body's mostly water. It's transparent. It's a complex mixture of macromolecules, especially glycosaminoglycans, also called GAGs, proteoglycans, and then multi-adhesive glycoproteins. Okay. Now getting into some of those components, those glycosaminoglycans, also known as mucopolysaccharides. Most of the time, 9 times out of 10, they're going to be called glycosaminoglycans. They are linear polysaccharides formed with repeating disaccharides. Di meaning 2, saccharide meaning sugar. So we have these two. We have uronic acid, which can either be glucuronic or iduronic acid. So we have a uronic acid, one of these two, combined with a hexosamine, which can either be glucosamine or galactosamine. Okay, so we have a uronic acid plus a hexosamine. Let me do this. So the disaccharides of GAGs equals a uronic acid plus a hexosamine. And then you take, so you pair one of each of those together, and then you combine a whole bunch of them together, and that gives you the glycosaminoglycans. They are covalently bonded to core proteins to form the proteoglycan, except for hyaluronic acid is not linked to a protein. Any kind of exception, a lot of times that's going to come up on tests. They like exceptions to the rules. Then abundant sulfate and carboxyl groups make molecules hydrophilic and negatively charged. So things you want to remember. Remember these guys, the uronic acid, the hexoamine acid, that they're turned into disaccharides, and then they bond to a core protein, which we'll see this in a second, to form the proteoglycan. And then remember that exception. So here are what our GAGs look like. They're long chains of polysaccharides made from disaccharides that pair over and over to form the chain. They are highly reactive to water, lots of water in the extracellular matrix. So here we have each of these examples. You have one of the two all binding together. And then these are going to continue to bind more and more, so they'll bind on this side to form a chain. I think this might be worded incorrectly. Let me... No, that's correct. Proteoglycan. Okay, it's worded a little bit weirdly. Okay. But actually, I'm going to... Okay. Talking about proteoglycans now. So we this kind of gets stolen. We put a chunk of proteoglycan in here, showing that there's a similarity between the glycosaminoglycans and the proteoglycans. Kind of weird that that's there, though, but it is what it is. Okay, the proteoglycans, separate from the GAGs. So proteoglycans are made up of two components. You have a core protein and then an attached glycosaminoglycans. Okay, the core protein is the backbone of the proteoglycan, and then the attached glycosaminoglycans, or the GAGs, complete the whole process. So we have our core protein right here, right? This is our core protein here in the purple. And then all these linear chains, these are our GAGs. So the GAGs bind onto the core protein, and then that process, then that turns them into proteoglycans. Here's some of those examples. All of these are just ones you're going to want to recognize the name, recognize specifically, you know, that they have sulfate in every single one of these. Sulfate, 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 sulfate. So that's kind of a trick to help you remember each of these. You don't need to memorize these specifically, just, you know, recognize the names to be able to identify them as like, hey, these are our GAGs that combine onto a core protein to form the proteoglycans. Okay. Talking about another component, we took it into the fibers component. We talk about collagen. So collagen is the name of a type of protein as well as a fiber. Okay, it is a family of 25 plus proteins. So there's a whole bunch of different proteins involved within collagen. We're mainly going to talk about the four first types, though. Okay, honestly, mostly types 1 and type 3. But there are a bunch more types. So we're going to go over the process of how collagen is actually synthesized, how we get it. But to form collagen, we have to make this precursor called procollagen. This is going to be some kind of test question where it's like, what is the precursor to collagen? Answer being procollagen. Some kind of question like that you're going to see worded in some way. Probably with more words, because professors like to add more words that are necessary, but it's fine. Procollagen, what is it? It is a soluble and a triple helix of 3 polypeptides. We'll see what that means in just a couple more slides. After secreted, it is converted to tropocollagen, which is insoluble, and it self-assembles to form fibrils and fibers. We're going to walk through kind of each of these steps through the next couple of slides. So collagen synthesis, there's a lot here. If it's easier for you than looking at this picture right here, I went ahead and I wrote out all of what is happening down in here so you can actually have it right there. I also found a pretty decent YouTube video that kind of walks through the generals of this process. It's a little bit more detailed than what we need to know, but it has some good pictures and some good things. If you want to take a look at that, go right ahead. So, kind of the step-by-step. I'm just going to do this quickly, as this is something that you're probably just going to want to put into Quizlet, write out a little bit, know the general steps. You don't need to know these word for word, but know the general steps. So first off, from the nucleus, we get mRNA that's formed for each type of an alpha chain. When we form collagen, we're going to build alpha chains. These alpha chains, we're going to get 3 of them, and then we're going to bind them all together. Okay? So we get an mRNA for each of the 3 different types of chains. We get a synthesis of the alpha chains of pre-pro-collagen with the specific peptides. Okay? So we have the mRNA with the alpha chain. Then we get the formation of pre-pro-collagen. From there, a big important thing that we need to remember. I don't recall this being tested on very much, but it is probably something that it could very easily make a test question. The formation of collagen is vitamin C dependent. So we need vitamin C. Need vitamin to make collagen. If you don't have vitamin C, you get a disease called scurvy. You'll talk more about scurvy in Biochem 2, as well as in a bunch of pathology courses. But no, you need vitamin C in order to complete the next couple of steps to go from pre-pro collagen into the actual stuff that you need. So I should clarify. This first one is the alpha chains of pre-pro collagen. So we make all 3 of the alpha chains. We use vitamin C to finish all of that whole process. And then we take 3 of those chains and we fold them all together to make what is known as a triple helix. This then goes to the Golgi complex. What does the Golgi complex do? It's the FedEx store. It packages it and sends it out to where it needs to go. It is secreted, and it's assisted by microtubules and microfilaments to get to the cell surface. And then it gets discharged out of the cell to then be used to form full-blown collagen. So reviewing this step by step, we make mRNA out of the nucleus, right? We copy the DNA signal, comes out of the nucleus, and we build a polypeptide chain, and that is the alpha chain. We build 3 different alpha chains, and then we combine those 3 different alpha chains. Once those 3 alpha chains are all put together, that's what we call pro-collagen. Okay? Yeah. There's actually one additional step here. Once they're all put together, once all 3 of them are put together, that's called pro-collagen. When they start to combine, but before they're fully combined, we call them, the components we call pre-pro-collagen. So when we have the components, but they're just starting to be put together, but they're not all the way put together to be formed into pro-collagen, we can call that pre-pro-collagen. Type of question that you might get asked would be something along the lines of, what is the precursor, what is the immediate precursor of pro-collagen? Good answer would be alpha chain, so 3 alpha chains. This is a good answer. The better answer would be pre-pro collagen. That's going to be our best answer. In case you could ask a question like that. Anyway, so once we have that pro-collagen formed, we talked about, thanks to vitamin C, it gets put into the Golgi complex. It gets taken to the outside of the cell. It gets exocytosed. It gets pushed out of the cell. And then it actually comes into here. These alpha chains, these come together. They form those triple helices. Those are then collagen molecules. We call that... There's our pro-collagen. Then those pro-collagen fibers combine all together. A bunch of those combine in order to form these collagen fibrils. Okay, this is when little details of words become super, super important. From alpha chains, we go to pre-pro-collagen, and then we go to pro-collagen. And then from pro-collagen, we combine a bunch of pro-collagen together to form collagen fibrils. And then we put a bunch of collagen fibrils together to form collagen fibers. It's collagen fibers. The word fiber is often also associated with the word cell. So you could call a collagen fiber a collagen cell. Later on, like, we'll talk about muscle fibers, and a single muscle fiber is made up of muscle fibrils, but the single muscle fiber is considered one muscle cell. So don't worry too much about that. Don't want to make your head spin. We'll get there when we get there, okay? So this is just another one of those processes, kind of like the chromosomes. When we talked about those, remember? It has each individual step going from smallest to biggest. We just need to sort out what that order is. So writing it all out, I'm just going to drop a new slide into here. Order of collagen formation. We start off with mRNA. That gets turned into the alpha chain. Alpha chains, 3 alpha chains are pre-pro collagen. Vitamin C helps and does its thing. And the 3 alpha chains all form together. That is now pro-collagen. Goes to the Golgi complex, gets packed and sent outside of the cell. Pro-collagen combines with other pro-collagen to form collagen fibrils. A bunch of collagen fibrils come together to form a collagen fiber. That's kind of the whole thing laid out, what you need to know. Now, actually talking about collagen itself, we have a couple different types. Like I said, there's 25 different proteins and stuff. We're going to focus on the first 4, mostly type 1 and type 3. But essentially, this slide right here, this is most of what you want to know, okay? Type 1 is ubiquitous, meaning that it's found everywhere in the body, okay? 90% of the collagen in the human body is type 1. These are also the largest and the strongest fibers, okay? These are what's really like grabbing on and holding us all together, that type 1 collagen. Type 2 is found in hyaline and elastic cartilage, and it is only fibrils, okay? So type 1 has fibers, type 2 only has fibrils. When we talk about hyaline and elastic cartilage, we think hyaline cartilage, that's the cartilage that's found in between our joints. So take like the knee joint, the elbow joint, any of those articulations in our body, you have layers of hyaline cartilage that allow the proper movement of everything. It's very, very fine. It's made of the fibrils. Type 3 is known as reticular cartilage. Reticular collagen. Reticular means net-like. You'll see why it's called net-like in a little bit. And then type 4, there's no fibers. It's a meshwork specifically in the basal lamina. This slide, 100%, pop each of these into a Quizlet, because like these kind of basics, these are the kind of questions that are going to get thrown at you in on the test. Each of these is going to be asked on the test at some point. Either this test, a lab exam, final exam, something like that. It's going to get asked about these different types and like the general characteristics of each of these. And we'll go a little bit deeper into specifically type 1 and type 3. So here's another little, table showing all of these different ones, all the different collagen types. Again, we don't need to know all of them. Main ones, you need to know 1 through 4. Type 1 collagen. Here's a picture of type 1 collagen. We have those big, big fibers. All this stuff in between is the collagen. we have those fibroblast nuclei, those fibroblasts, making the collagen, doing that kind of stuff. Clarify, this cell is collagen synthesis. This cell right here, this is going to be a fibroblast right here. Insert, text box, fibroblast. I love making these videos because it makes me come back to all this stuff so much and really double check and question everything. So, plus you get a lot of like the extra tidbits as I record this as well. Okay, let's try and fly through the last little bit. Type 1 collagen. So type 1 collagen, these are responsible. I'm going to say type 1 collagen is responsible for the strength of the extracellular matrix. High tensile strength, these are the big ones. Collagen is made by those fibroblasts. Good, those are the big things you need to know about type 1. Type 3 collagen, a couple things I'm going to show you that for sure are going to be asked. These are very thin. Remember the type 3. I didn't actually say whether they're fibers or fibrils, but actually just right here, fibers. These are very thin fibers. That tends to be asked. It tends to, again, reticular means net-like. That's typically asked as a test question, which of the following is a net-like, something like that. You have high affinity for silver stains, are gyrophilic. That, again, often a test question, whether they're in lab, because this is what that looks like. All the black, that's the silver staining. So you'll see this is the same thing in lab. And the test question is going to ask, what type of collagen is this? Or it's going to ask, what staining was used to do this type of collagen, or this type of stain? And silver stain, aka argyrophilic. It is found in lymphoid organs, such as the lymph nodes and the spleen. It's also found in structures like blood vessels, uterus, and the bowel, where you need elasticity. So type 3 collagen is very elastic, which means it's very flexible. Elasticity means that it has the ability to return to its shape after being stretched. So like blood vessels, the uterus, the bowel, once it's been stretched, it bounces back. It comes back to the form that it had before, and that's the job of type 3 collagen. Fiber bundles are composed of elastin, and cross-linked peptides provide extensibility. There's, again, that picture, those elastic fibers. Here they are relaxed, and they all get stretched out, and they cross-link, and they detach to each other, and they go back to their original shape. There's another potential test question, more on the lab side of things, is you need a different stain. So you can't see the reticular fibers, like the silver fibers like this, on the same stain as the elastic fibers. So these black guys right in here, these little black lines, those are our elastic fibers. And you have to use a different type of stain for those, because those are not going to show up on this type of staining, because they're slightly made of a slightly different material, so they're going to attract different stains. Now, adhesive glycoproteins, we've mentioned fibronectin and laminin before. We're just going to go over quick their jobs and a couple of the fun fact details on them that could be on the test. So fibronectin binds cells to components of extracellular matrix. It is also a binding site for cells, collagens, and heparin. Laminin is synthesized by epithelial cells, and it binds those epithelial cells to components of the basal lamina, and also has binding sites for collagen, heparin, and cells. And I'm going to adjust this so that way it's the same as above, because that makes more sense. So this is the same for both of them. Laminin synthesized by epithelial cells and in the basal lamina. That's usually what's asked, that the laminin is in the basal lamina. Laminin is in the lamina. The fibronectin structure. So each fibronectin subunit is folded into a series of rod-like domains. Here are all of the rods, domains where different things can bind. Several domains bind one or more extracellular matrix macromolecules, including, collagen, heparin, and fibrin. Other domains recognize and bind cell surface receptors via what is known as the RGD sequence. Don't worry too much about what this is and how it all works. You just need to know what it is. The RGD sequence is an arginine, glycine, and aspartate sequence of amino acids, which allows for binding of cell surface receptors. Okay, that's all you need to know. Don't try and go too deep into that. Just know that this sequence, that RGD sequence, allows it to be a cell surface receptor. Okay, that's all you need to know. So here we go. Fibronectin has 2 identical polypeptide chains. These are those rod-like domains. Each rod-like domain is prepped to bind to different things. Also, it's bound by disulfide bonds. That likes to be a test question as well. So rod-like domains, the RGD sequence. Good. Fibronectin is also involved in cell movement. So when migratory cells are grown on fibronectin, they adhere to it. In the embryonic cell migration, the pathways followed by migrating cells are rich in fibronectin, suggesting it may guide the cells. And also many kinds of cancer cells do not produce fibronectin. So because they don't have that fibronectin, they can't move properly and anchor properly, so that it kind of connects into metastasis. Give me 1 second. Okay. Next protein, laminin. Laminin is made up of 3 polypeptide chains, 1, 2, and 3. It is a binding site for collagen type 4. Thanks. Snacks are important. Always have snacks. So there's 3 polypeptide chains, 1, 2, 3. It has a binding site for the collagen, specifically type 4 collagen and heparin. So you can see heparin binding domain right there. Heparin or heparin sulfate binding domain, collagen 4 binding domain, all these different places to bind and grab onto all of this other stuff. And then again, this one exists in the basal lamina. They are found mainly in the basal lamina. That's what we need to know. And then what the basal lamina is a thin sheet of specialized extracellular material. So it's the thin layer that all of the cells grab onto to stay in place. So we can see here it goes in between the connective tissue and the epithelial cell layer, the basal lamina right in through here. All of this stuff is grabbing on in between right here, combining onto the basal lamina in order to stay connected. So we have, connective tissue reaching, components of connective tissue reaching through, grabbing onto stuff of the epithelial cell, and stuff within the epithelial cell reaching down below, grabbing onto the basal lamina to keep everything anchored to where it's supposed to be. Integrins. These are cell surface receptors that bind some of the components of the extracellular matrix. There are a large family of cell surface receptors that bind to fibronectins or laminins and collagen. Integrins integrate or combine the cytoskeleton with the extracellular matrix, and it plays an important role in regulating cell movement and attachment. A lot of this is honestly just kind of slap this into Quizlet and memorize, okay? Because when exam time comes around, specifically, you just need to kind of regurgitate some of this stuff. So integrins are cell surface receptors that bind to fibronectins, laminins, and or collagen. Integrate the cytoskeleton with the ECM. Test question possible right there. And then also regulating cell movement. So not a lot you can do as far as like understanding integrins, just straight up know those little components. They're plasma proteins binding site for extracellular matrix components. And this is what they look like. So they go in through the cytosolic membrane, outer membrane, to be able to grab onto stuff and hold and anchor. There is our integrin protein. And if I'm not mistaken, we talked about integrin proteins as well previously when we talked about plasma membranes and kind of the components of plasma membranes. So it's coming through, grabbing onto the fibronectin, grabbing down into the cytoskeleton, integrating the cytoskeleton to the extracellular matrix, keeping everything attached. Now, talking specifically about the basal lamina, it's that structural support and a permeability barrier. So there's the epithelial cells, the collagen fibrils, also like that cytoskeleton, that connective tissue, the basal lamina in between, and it supports the structure overall. Specialized extracellular matrix attaching epithelia or epithelial cells to connective tissues, bunch of type 4 collagen fibers in the matrix, and it has laminin. And there's just another picture of some of those type 4 collagen fibers, laminin, and some other proteins. And that is the extracellular matrix. Like, hopefully we've seen, a lot of these words before. So it's just kind of continuing to see them over and over and over again. And I encourage you, as you go through this material, say the things out loud. The more that you say the big words, the different proteins, the chemicals, especially in biochemistry and microbiology next trimester, microbiology and immunology, the more that you can say the words out loud, say them out loud along with me, Sometimes I butcher them, that's fine. Look up how to pronounce them, that's great, even better. The more that you can start to use these words as part of your day-to-day, language, maybe not every single day that you're going to use this, but you can read a test question and your brain doesn't just gloss over the word. Instead, you actually read it. You're going to be able to understand a lot better this material once that the words kind of integrate and become part of your brain. You're going to be in a much better position. And of course, again, thank you so much for watching. Go ahead and leave a like on the video. If you have any questions, comments, leave those down below as well, and I'll make sure I get to them for you. And of course, if you're looking for my notes for all of this material, it is going to be down below in the description. You can find the link to get the access to all of this. And have a wonderful night, and good luck studying.